morning everyone. In the chapter of reproduction in flowering plants, in today's tutorial we are going to study about the male reproductive part. So first of all we start with the structure. The male reproductive part, which is also known by the name stamen, and if we draw the structure of the male reproductive part stamen, a group is also called as androecium. So generally there is a lobed structure, and to this lobed structure there is a long stalk like structure like this, and this is the general type of structure where this part is known as anther. As you can see here in this diagram, we have shown two lobes to it, and this long slender structure is called as filament, and the whole structure is called as the stamen or the androecium group name. Generally the anther is bilobed, and this type of anther is called as dithecus. This is a general type of arrangement where the two lobes are connected by a sterile tissue which is called as connective. This is what is called as connective. Some plants like Malvaceae family, they have a single lobed anther like this. There will be one lobe only, and to this one lobe there will be a stalk of filament. This is called as monothecus. Mono means single and thecus means lobe. If there is a single lobe, it is monothecus. If there are two lobes, it is dithecus. And monothecus example we have mentioned earlier, it is family Malvaceae. Right. Now there are different positions of the filament with the anther. If we show it on the board, so the structure will go like this. We will be drawing the bilobed structure always, because it is the most common type of anther. So if it is at the base, this is called as the basifixed position. And suppose this filament is on the dorsal side. So how it will be on the dorsal side? So here is the dorsal surface. So if it is like this, so this type of position is called as dorsifixed. And the anther may be on the whole lateral surface here, like if we show it here, what is like this on the lateral surface, then this is called as adinate position. And there is one more form here, and for this we will be drawing the anther first, bilobed structure, and the filament is in the center where the anther is able to dangle. This is seen in the case of anemophilous flowers. Those flowers who are pollinated by air, they are called as anemophilous, and this type of position is called as versatile position. So there are different positions of the anther with the filament. It could be basifixed, dorsifixed, adinate, or versatile position. Now coming to the detailed structure of the anther and the filament. So for this purpose, what we are going to do, we are going to draw the anther first of all. So this is the anther, and again it is a bilobed dithecus anther, to which there is a filament connected like this. The part of the filament towards the anther is this one, is called as the proximal. and this part of the filament here is called as distal. The proximal is always connected to the anther and the distal is to the any floral part. It could be to the petal or even to the any other floral pore. And the detail of anther will be seen when we see the transverse section TS of anther. And the diagram which we will be drawing will be for a diathecous anther. So TS of a diathecous anther. So in this, what we find here, that if the anther, this is the anther bilobe diathecous, and if it is cut from the transverse section, now the cut surface will be visible in this form. To show that structure, I have to rub it. The cut surface we are going to see here. It will be visible in this way. This is the outline for the cut surface, and here this is the first lobe and this is the second lobe. This is the first lobe from here to here, and this is the second one from here to here. Each lobe have two compartments, and these two compartments are actually the microsporangia. These are pollen sacs which we will be drawing in this manner. This is the cut section. Right. Each lobe have two microsporangia which later on develop into pollen sac at maturity. So these are the four microsporangia, and the region from the microsporangia to this outer surface is called as, this part is called as the anther wall. Anther wall. And this anther wall is made up of different types of cells and tissues. Like the outermost surface is made up of flat cells like this. They are flat cells, and these flat cells don't have any intercellular space, and this is called as the epidermis, sorry. And to this inner one there will be some radially arranged cells, and these radially arranged cells are in the layer of 2 to 3 layers. There are 2 to 3 layers of radially arranged cells, and this is called as the endothecium. Inside to endothecium there is parenchyma cells. Loosely arranged parenchyma cells are there, and these parenchyma cells are called as the middle layer. And these three, epidermis, endothecium and middle layer, they form the anther wall. The outer lining of this microsporangia is made up of pyramidal shaped cells, and these pyramidal shaped cells which are lining the inner lining of the microsporangia, this is called as tapetum, and this tapetum is responsible for providing nourishment, because inside there are archisporial cells present inside. We are talking about the immature microsporangia. There are archisporial cells which later on develop into microspore mother cells at the time of microsporogenesis, that is the formation of microspores. So the tapetum provides nourishment to these cells. These tapetum are having dense cytoplasm, and they are generally multinucleated. Along with that, they are responsible for the formation of sporopollen in the pollen grain and pollen kete formation. So these are the layers of the anther wall, and here in between there is the connective sterile tissue. So this is the diagram which you will be able to find in the transverse section of a anther. This is all for today, and next topic we'll be discussing about the process of microspore formation, that is microsporogenesis. Till then, thank you.